Okay, we're going to go through genetics, the fun stuff of how do we know that things make the things that they make and how did we learn that, etc., etc. So, genetics. We start our genetics discussion with a man named Mendel, Gregor Mendel. He was a monk, and he did a whole bunch of experimentation with pea plants. In fact, that's what he did. He was a monk. He lived in a monastery, and he was in charge of the gardens eventually. And he started noticing things about the pea plants he was working with, and he just started doing what ended up being genetics testing on these pea plants. And that gave us the basis for a whole bunch of information, and as we know it, for genetics. So modern genetics begin in the mid 1800s, that would be the 19th century, in an abbey garden where a monk named Gregor Mendel documented inheritance in peas. And once again, you have to remember, we only know things happened because he documented it. He wrote everything he did down. He was meticulous in his notes and his numbers and what did what and what he mixed with what and whatever. And so he used experimental methods, which means that he started by noticing a phenomenon, and then he came up with a hypothesis that the reason this is happening is because of this. And then he did experiments on it and saw does his data support his hypothesis or does it not support his hypothesis, and he has to continue on with his testing until he gets a conclusion. He used quantitative analysis, which means that he, used numbers. He quantified. Everything had a count. It's an excellent example of scientific method. So he bred pea plants. The good thing about pea plants is you're working with a very small genome. And the other good thing about pea plants is that if you cross for only one trait, which is what's going to be important in Mendel's work, you are clearly going to see the results. So how did he breed pea plants? I'm put all the words up here. Actually, maybe I should wait. He did something called cross-pollinating. We'll start there. He cross-pollinated. Cross-pollination occurs normally when any kind of wind or insects take pollen from one flower to another, but he can be very absolutely certain where his pollen is coming from. And so what he actually did, he took true breeding parents. And what a true breeding means, so here are our two parents, is that this parent will always give white flowers and always gave white flowers and always gave white flowers. And this parent always gave purple and purple and purple. And he wanted to see what happened when white pollinated purple. But how do you make sure that the pollen is coming from the white flower? So pollen comes from stamens of a flower. A stamen is what makes all the pollen, and the pistil is what has the ovary space for the flower. So it's the male and female parts. You can cut off the stamens of a flower to make sure that pollen isn't going anywhere and make sure you are only getting pollen from a very specific flower or plant. So that's what he did. He made sure that pollen, he absolutely was certain that pollen could have come from this white flower, and he pollinated the female part or the stigma of this purple flower. So stamens and anthers, you're right, stamens and anthers. The stamen is the stalk part, and the anthers where the pollen is. My apologies. Stamens and anthers, stigma and pistil, same, whatever. Plant anatomy. You learn about it in 1407. Then you get babies, except they're not like baby babies, but they're pea plants. So you get an F1 generation. F stands for familial. So this is a family of plants that is one generation removed from the parent plant. And we see that only purple flowers result from this crossbreeding of a white flower to a purple flower, okay? Then you can self-pollinate your F1 generation, your familial or filial generation. So you make sure that this purple flower sibling only breeds with this purple flower sibling. And don't be fooled, plants can also some come to inbreeding, and you get weaker plants and plants that don't produce as much as well. But for the work of Mendel, this was very important that he did crossbreed the sibling plants because he needed to see what was going to happen in each generation. And then you get an F2 or filial generation. And it's in his F2 generation that you see what happens to each dominant or recessive trait of a pea plant. And so throughout his life, Mendel collected data for 7 pea traits, and he did this by only breeding dominant forms to recessive forms for whatever trait he was looking for. So for every trait he was looking for, he made sure to cross a true dominant to a true recessive plant. It's very important for our purposes of understanding how his genetics worked. So he did flower color, he did seed color, he did seed shape, he did pod color, he did pod shape, he did where the flowers were growing, if they were growing only at the very ends, oh, I'm sorry, that's terminal, if they were growing at the very ends or along the whole vine. And he also checked for vine height, if it was a tall plant or a dwarf plant. So these are all the things he did throughout his lifetime. I'm sure he did more, but these were the 7 that followed along with each other that became very important for our understanding of genetics. So let's look at a parental F1 and F2 for. As we've already shown, when a true breeding white flower, so a white flower that always gives white flowers, is mated with a true breeding purple flower. So it's a purple flower that came from purple flowers that came from purple flowers. Probably even more generations than that, but we'll just stick with 3. In our F1 generation, we see we get 100% purple flowers. That tells us that purple is the dominant color trait in Mendel's or in these pea plants. But what happens now if we do an F2 generation? So self-pollinate your F1 generation and looky here. In our F2 generation, we are going to get 75% purple and 25% white to a ratio of 3:1. Remember the ratio 3:1 for an F2 generation of true breeding dominant and recessive parents. In all of our studies since Mendel, if you have a true breeding dominant and recessive parent in your F2 generation, you will always get a ratio of 3:1 in your color. So what happened? Where did the white flowers go? Well, we now know that we would use the term that they are recessive, which means that this is going to be big P, big P. Wait, I can actually draw on this, can't I? Okay, this is big P. Make it a big P. Big P. And this is little p. I'm going to give it a squirrely so we know it's little. And little p, right? If you cross a big P to a big P, little p, you have 100% big P, little p. I know it looks like a Q, just go with it. You have 100% big P, little p. I'm getting way ahead of myself in this PowerPoint, but I just want you to see how we get there. If you cross 100% of big P, and little p, I'm sorry, just know that that's a little p, then what's going to happen is you're going to get 2 big P's here. You're going to get a big P, little p, which we already know shows as a purple. You get another big P, little p. I know this isn't perfect, just go with it, that we already know shows as a purple. So there's our 75% purple. And then we get one box that has a little p and a little p. And there's our 25% white. And it doesn't matter what trait you are looking for, it happens in that ratio for every true breeding parent that we have ever tested. So that's wonderful. You already know that if you have a true breeding purple and a true breeding white or a true breeding whatever and a whatever, your F2 generation is going to come up with a ratio of 3 to 1. Sorry, let me clear, undo. Let me just clear. Okay, erase, fine. 3 to 1, there it is. Offspring. Excellent. That's one of the major things you need to know in this PowerPoint. And so you see the white flowers came back. Oh, this doesn't, I thought it would go away with that slide. You can see how much I've used that tool. Okay, so what does this mean? Because you've been through mitosis and meiosis, yes, you did it on your own, but you've been through it. You know all about how traits can come in alternate versions and how in meiosis, those alternate versions can cross over and form tetrads and move into different chromosomes, and we get all sorts of different traits because of that. But Mendel didn't know that. So he, in this case, was looking at purple versus white flower, and I don't believe that he is the man that came up with the word alleles, but we came up and through history and genetics with the word alleles, which is saying a different allele or different alleles vary in the sequence of the nucleotides, so A, T, G, C, and DNA, at a specific locus of the gene. So here is an allele for purple flowers, and here is an allele for white flowers. And thanks to the whole process of crossing over in meiosis, you can get different variations of purple or white or purple, and we know in the F2 generation that's going to be 75 to 20 or 3 to 1. So the purple flower allele and the white flower allele are 2 DNA variations at a flower color locus. And different versions of gene at the same, they're different versions of a gene at the same location on homologous chromosomes. We know that purple masks white. We know that every offspring of a genetic pair has one of each, a mother, I guess, and a father, if you will, allele. So it just depends on if they get a dominant allele, 2 recessive alleles, a dominant and a recessive allele, what they're going to look like. Get these all up here. Nope, let me go back. So, as I just mentioned, we know that traits are inherited as discrete units. And for each characteristic, an organism gets 2 alleles, 1 from each parent. This is what a diploid organism is. You are a diploid organism. Every somatic cell in your body has 46 chromosomes. The only cells in your body that have 23 chromosomes are sperm if you are a biological male unit, or eggs if you are a biological female unit. And they get that, so they become haploid. So they're 23 and 23, so that when a sperm and an egg meet, it makes a 46 chromosomal embryo, and that becomes a whole new human being. So we inherit 2 sets of chromosomes, 1 from each parent. They are homologous chromosomes. You're getting the same information just from each parent. That's more true for females when we get the XX, because we are getting the exact same amount of genetic information. For men, the X chromosome that you get from your mother holds a whole lot more genetic information than the Y chromosome that you get from your father. And I'm not even saying that to be like rude. That's just, the Y chromosome is actually a very, very small chromosome. But there are still homologous alleles or chromosomes. They're still, there's too many words. There's so many words. There's still sister chromatids, complementary alleles, whatever you want to say, between the mom and the dad. So it's like having 2 editions of an encyclopedia. You have Encyclopedia Britannica, that would say that's from your mom, and it has all the information that you need from your mom, and then Encyclopedia Americana from your dad, and it's all the information you need from your dad. What are the advantages of being diploid? I'm going to pretend you all answered me, is that if you have a chromosome that is recessive in a bad way, such as a genetic mutation or some sort of something that is life-ending, you have another chromosome to cover for it. It's basically a safety net. So one of your homologous chromosomes for a trait can be completely mutated and unfunctioning, but if you have a dominant chromosome that is still intact, then you're going to be fine. The issue might come if you have kids, or if you have, more so in your grandkids is where the issue might really come up. But yes. So we now know dominant recessive. I keep using those words. I'm using them too early in the PowerPoint, but there's no reason to sugarcoat it. We found that some traits mask other. The dominant purple masks the recessive white. They're different flower colors. They are not blended. Now, this does not mean that flower colors don't come along that do blend, and that's a whole different genetic phenomenon. But in terms of dominant and recessive, there is no blending. You are either purple or you are white. That's it. That's all you get. You don't get a light purple. The dominant allele is the functional protein. So it's the protein that is being expressed. And when the dominant allele is being expressed, it masks the other alleles. The recessive allele makes a malfunctioning protein and or doesn't make a protein at all. And so if you have two recessive alleles, that's when you see the secondary trait come out. Okay. I was told that when those noises are happening, you can't hear me speak, so I am trying to patiently wait for all of them to go away. Wild type is the allele producing the functional protein. Mutant is the allele producing or not producing at all the malfunctioning protein, and those are homologous chromosomes. Okay. So now we have to look at genotype and phenotype because they are two different things. It's the difference between how an organism looks, what is being expressed, and what its genetics are saying. So the phenotype is how you look, and it's what is the protein that is being expressed. The genotype is your genetics, A, T, G, and C, and the order that your A, T, G, and C's fall in your genome. So we're supposed to use the words phenotype and genotype and dominant and recessive, explaining Mendel's results here. So the dominant purple, which is expressing a phenotype purple, is dominant to the recessive white or the phenotype white. And the F1 offspring are all heterozygous, but they're expressing the dominant purple phenotype. The genotype is what happens when I have to draw big P's and little p's. Now, big P and little p, of course, being total simplistic version. But in a purple, it's big P, big P. That's the genotype. And the white, it's little p, little p. Come on, Curly Q. And in the F1 generation, we do have a big P, but we also have a little p, one from each parent. So we have a homologous big P to a homologous little p. The big P is dominant, though. That's the expressive phenotype. And so all of them are going to be purple. All righty, Roo. So what I'm doing is expressing genotypes as letters. I'm choosing arbitrary letters to show what the alleles are for the pea plants. You typically use the first letter of the dominant trait, just making life easier for everybody. So big P is purple, little p is white. We know. Oh, and let me throw in other words here. Let me see. I can text, right? Yes. So this right here, these are homozygous or homogeneous. No, it's just homozygous. These are homozygous, and so is these. So that means that they are the same. Homo means the same. Hetero, these are heterozygous here. So you actually have different alleles, big P and little p. Homozygous big P, that's homozygous dominant. Actually, let me throw that word in there. Dominant. And then little p is homozygous recessive. That is literally how we would explain things. And then you have heterozygous. Heterozygous cannot be dominant or recessive because it's both, right? Okay. I hope y'all are with me so far. How come I haven't used these annotating tools the whole time? It's a good question. It's a good question. So just to repeat a whole bunch of what I have said, your F1 generation, your homozygous dominant, these are your phenotypes here. They're true breeding purple and true breeding. The genotype is that they are homozygous dominant and homozygous recessive. In your F1 generation, good Lord. In your F1 generation, the phenotype, of course, is that they're all purple flowers. The genotype is that they're heterozygous, big P, little p, big P, little b. After we self-pollinate our F1 generation, after we self-pollinate our F1 generation, our F2 generation is not going to be all of one thing or another. It's actually going to start having different genotypes. So you're going to have, so what are these different genotypes? So the phenotypic ratio, it's very important. It's very important. It's very important that you know this. Phenotypic ratio is 3:1. The genotypic ratio, just to throw it out there, because you're going to need to know these ratios for your tests and everything, is 1 to 2 to 1. Now, this is true regardless of what you're breeding for, as long as you have a true homozygous dominant and a true homozygous recessive as the parents. Your F2 generation should always show a 3:1, and it should always show a genotypic ratio of 1 to 2 to 1. What do I mean by 1 to 2 to 1? I already showed y'all, and that means the next few slides are going to show you again, but one of your offspring is going to be homozygous dominant. 2 of your offspring are going to be, little p, heterozygous, and 1 of your offspring is going to be homozygous recessive. So let's see that in action. And how do we do that? We do it with a Punnett square, Punnett square, Punnett square. Everybody loves a Punnett square. Okay, maybe you don't love a Punnett square, and that's fine. But that's what we use. And typically, they're easy-ish enough to use. When we get into the 16 Punnett squares, it gets a little hairy, but they still have ratios. So this is our F1 generation, not our parents. So the male, the pollen, which is in sperm, pollen is sperm, by the way, is a big P, little p, and is a big P, little p. And so let's look at our F2 offspring. Big P, little p, big P, little p, little p, little p. And here is where we get the phenotypic ratio of 3:1. 3 purple to 1 white. So that's phenotype. And so genotype is 25% big P, 50% big P, little p, and 25% little p, or 1 to 2 to 1. And there's our 3:1. So yes, I've said it like 16 different ways in the last few slides because slides keep repeating themselves. But the reason it's repeating themselves, it's because it's important that you know the phenotypic ratio is 3:1 in the F2 generation of true breeding parents, and the genotypic ratio is 1 to 2 to 1. That's when we are only solving for one trait. It gets a little crazy when you're solving for 2 or 3 or 4 or 16 or whatever. But this is for 1. This is true for when solving 1 trait. Okay, that's when that is true. And it'll always be true when you are solving for 1 trait. Let's move on. Oh, yes, well, yes, phenotype and genotype can have different ratios. We've already used these words, but we have homozygous, which is the same alleles, and it can be homozygous dominant or homozygous recessive. And heterozygous is when we have our different alleles, so a dominant and a recessive. But look, homozygous dominant and heterozygous both give us purple because the purple is dominant. It's only when we hit homozygous recessive that we see the white. Two organisms can have the same phenotype but have different genotypes. As I just said, homozygous dominant and heterozygous both give us purple. So how do you know what the phenotype is of the purple? Is it a dominant purple or is it a heterozygous purple? You have to crossbreed it. You can't tell just by looking at it. So you breed the dominant phenotype with an unknown genotype. With an unknown, so you breed the dominant phenotype, is it big P, or big P, little p, with a homozygous recessive. So little p, in this case, a white flower. And that will tell you what the hidden alleles are for the dominant flower. If this is big P, if the purple is big P, then you know that what you're going to get out of it is 100% purple. If it's big P, little p, well, the ratio changes. We call this a test cross because we're trying to test what the alleles are. Oh my God. So is it big P or big P, little p? Okay. If it's big P, we already know, we've seen it. You're going to get 100% purple. But if it's big P, then you have big P, little p. Well, those are going to be 50% purple. But then you have little p's across the board on the bottom, and those are going to be 50% white or 1-to-1. Depending on what results you got, you would know what the parent unknown was in terms of genotype. So this leads us to Mendel's first law of heredity. First law of heredity. What did I say? And that's the law of segregation. The law of segregation is that during meiosis, alleles segregate. And each allele is packaged into a separate gamete. And so it's really just the luck of the draw if you're going to get a dominant allele or a recessive, or one of each. So remember, you're going to have two parents. So we just did meiosis really quickly there. And now you have 4 separate gametes here. And it's just the luck of the draw. Which gamete are you? Which sperm or egg are you in this picture? Are you the second one? Are you the fourth one? Nobody's going to know until you're born what you are. This happens during metaphase 1 of meiosis when this law of segregation occurs. So yes. Some of Mendel's experiments followed the inheritance of single characteristics. Most of them did. Seed flower, color flower, those are those mono-hybrid crosses that are only the 4 squares, the easy ones. But he also wanted to know seed shape and seed color. Those are 2 different things. Seed shape and seed color are 2 different alleles. And so then you are solving for more than just one trait. It's more difficult. So these are called dihybrid crosses. And remember, Mendel is doing all this without knowing about DNA or genetics or alleles or chromosomes. He doesn't have that background and vocabulary in his head. He's just looking at pea plants. So you gotta understand, it's fairly impressive he was able to do what he could do with just looking at pea plants. It's like going back to, and here I'm going to show my lack of scientific knowledge, whoever first said that the earth revolved around the sun. Because until somebody said that, the thinking was that the earth was the center of the universe, because people are that self-centered, and that everything revolved around us. And so the guy who said that, no, we go around the sun, I'm pretty sure he was killed for his thinkings. but he was right. And he was able to figure it out long before we could do anything to prove it. So all the things that people could figure out before the science caught up with it, it's just really, 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 really interesting. And I hope it fascinates you and you'll look into the history sometime. Okay, let's talk about a dihybrid cross. God only knows how many noises this slide is going to make, so I'm just going to try to. Okay, we'll stop there. Okay, a dihybrid cross is 2 traits. You're looking for seed color and seed shape. So here we have dominant yellow and dominant round. So a yellow round seed. It's 2 different traits. They're 2 different alleles. They're in 2 different places on the genome. So you have to have the dominant for both allele. So it's 4, okay? And he is going to breed that to a true breeding green, little y, and wrinkled little r, p. Remember, true breeding yellow round is the phenotype. Big Y, big R is the genotype. True breeding green wrinkled is the phenotype. Little y, little r is the genotype. Our F1 generation, because we have a dominant, a homozygous dominant for both traits, we are going to see 100% phenotypic yellow round peas. But since we are doing true breeding crosses, we know that every single one of these seeds, or peas, a pea is a seed, by the way, if y'all didn't know, is big Y, big R, little r. If you need me to prove it, I will do so. So 1, 2, 3, 4. Let me just do this real quick. Big Y, oops, big R, boop, 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 little y, little r, little r. My drawing skills are not. So it's just a Punnett square, but now you have to do it 4, 8, 12, 16 times. Big Y, little y, big Y, little y. All the Y's are big Y, little y, big Y, little y. And all the R's are big R, little r, big R, little r. Okay, so I really did that wrong. I should have just separated it in 2 small Punnett squares because you're going to get little y, r, but that doesn't mean anything. Right, I did the second cross. I wonder if I can erase this part of the presentation. Okay, let me see, let me erase this. Okay, and I can prove it. I'm going to do it easy way. I'm going to do it an easy way. No, give me my drawing tool. Give me my drawing tool. Okay, I don't know how to do things. You? No. Okay, what's happening here? There it is. I'm going to do it the easy way where I'm just going to separate my traits. You can do this. This isn't going to kill anybody. And you see that for every single one of these, you're going to get big Y, little y. And if I separate the R's, for every single one of these, you're going to get a big R, little r, which means you're going to have 100% big Y, big R, little r. Okay. All right. But what happens when we self-pollinate? Sorry. What happens when we self-pollinate this one generation? We are going to see 4 different traits come out. Because remember, we are actually testing for 4 different traits. Is it yellow? Is it green? Is it round? Is it wrinkled? That's 4 different things we're testing for. So you're going to get 9/16 of them will be dominant yellow round. 3/16 will be green round. 3/16 will be yellow wrinkled. And 1/16 will be green wrinkled. A ratio of 9:3:1. This ratio is true for two traits that are true breeding dominant and true breeding recessive from the parents. So this is a true ratio for two traits. Remember, it's 3 to 1 for one trait. For two traits, it's 9, 3, 3, 1. So what's going on here? If genes are on different chromosomes, they sorted together or independently in the chromosomes. So are you getting color and shape together or not? What? Oh, I'm sorry, I see what they're doing. Are you getting all dominant together and all recessive together, or are you getting them in all sorts of ratios? Well, the answer is, well, here. If you were getting them as all dominants or all recessives, this would be your ratio here. We know that's not what happened because we got 9, 16, 3, 16, 3, 16, and 1, 16. So that's not how we did that at all. So there's the little death screen. So we got to do a dihybrid cross. Ah, there it is. Oh, yes. Oh, it never ends. The happiness never ends. And so a dihybrid cross is taking every single formula you can from your two alleles. Is it dominant, dominant? Is it dominant, but then recessive? Is it recessive, but then dominant? Or are they recessive, recessive? And crossing them together. And when we do this, we see that we get 1, 2, 3, 4, 5, 6, 7, 8, 9 yellow round, excuse me. We get 1, 2, 3 green round. We get 1, 2, 3 yellow round or wrinkled. And then we get 1 green wrinkled. And so yes, they are sorted independently and randomly in your gametes. So this is Mendel's second law of heredity, the law of independent assortment. The different genes or loci, which is what a gene is located at, separate into the gametes independently. So color is separating independently of So non-homologous chromosomes align independently. Classes of gametes are produced in equal amounts. The amount of dominant is equal to the amount of dominant recessive is equal to the amount of recessive dominant is equal to the amount of recessive recessive. But you're doing two traits. This is only true for genes on separate chromosomes or on the same chromosome, but so far apart that crossing over happens frequently. So at the very, very opposite ends of those chromosomes. And metaphase 1 does independent assortment. Exception is that the genes are really, really close together. They are usually inherited together. They are linked genes. Anyway, you've already had to do mitosis and meiosis, so what have you, but because of the random assortment and because of the law of segregation and the law of independent assortment, we end up getting the ratio of 3, 1, 1, 3. But remember, Mendel did not know any of this. This was not a thing Mendel knew. He was looking at peas, and that's what he found out. Okay, so, right. Law of segregation is best shown as a monohybrid cross when we are looking for a single trait. Each allele separates into separate gametes, and they are randomly separated into separate gametes. This is established by metaphase 1. The law of independent assortment deals more if you are looking at 2 or more traits. The genes on separate chromosomes assort into gametes independently, and that is established by metaphase 1 as well. The only exception of that is linked genes, which is not something we're really going to be concerned about right now. Mendel got lucky that he was working with pea plants. I mean, like they were in the garden, but he had a full vegetable garden and flower garden and herb garden. He could have picked any plants. He was just he happily chose peas. They're available in many varieties with distinct heritable features, such as flower color, seed shape, etc. And he had very strict control over which plants made it with what, by being able to cut off the stamens of the pea plants. He didn't want cross-pollinating. Not all plants are easy to do that with. Many of them aren't. The stamens are fairly hidden, or you can only get insects in there to get the pollen or whatever. So he happily chose a flower where the stamens were, and anthers, were exposed enough that he could just cut them off. Each pea plant has male and female structures. They can self-fertilize, but he could also cross-pollinate plants the way he specifically wanted to. They're also relatively simple genetically, and most characters are controlled by just a single gene. That was a happy accident that he was able to choose that, because, you know, plant height is just one gene. Plant color was just one gene. Seed color was just one gene. It wasn't controlled by multiple genes. Those are polygenetic things that we'll talk about next PowerPoint. And happily for him, one gene was always completely dominant over the other. He didn't have to deal with blending. Oh, that went quicker than I expected. I hope I didn't confuse you too much. The thing that's really important to know, are the ratios, the phenotypic ratios for the F2 generations on if it's a monohybrid or dihybrid cross. If it's a monohybrid cross, the ratio is 3:1, phenotypically. If it's a dihybrid cross, the ratio is 9:3:1. Genotypically, for a monohybrid cross, you get a 1:2:1. Phenotypically, for a monohybrid cross, you get, genotypically for a monohybrid cross, you get basically 16 different genomes. So that gets a little bit more dicey. It would also be very important. It's also very important for you, your homework and your understanding, for you to understand how that dihybrid cross worked, because I'm not hitting share. So, no. So this slide, no, sorry. This slide, showing how the dihybrid cross actually works, and it's 16 squares when you do a hybrid properly. You have to pull one allele, one allele from each of your choices here, but only do one parent at a time. So for example, we're not looking at this parent right now, even though they're exactly the same, okay? So just looking at this parent, you can have big Y, big R, boom. You can have big Y, little r, boom. You can have little y, big R, boom. Or you can have little y, little r, boom. You have to pull out every single possible combination of the alleles that you have. You're looking for the combination of the traits. You're not looking for big Y, little y. You don't inherit the two separate color genes together. You're looking for color and shape together. Color and shape, color and shape. So you look at all the possible ways that could happen. And then if you then do it for the other parent, and in this case, they're exactly the same, that's how you set up the dihybrid cross. And then you can just write down your answer. Okay. And because we are on the slide, we can just see the visual of what each of these answers mean. But notice here in your genomes, you have only one big Y, big Y, big R, big R. And then all the others that are dominant yellow and dominant round, they have all different sorts of genotypes. So we're not going to worry about the genotypic ratio of a dihybrid cross, but we are worried about that phenotypic ratio of 9:3:1. Okay, so you're not pulling big Y, big Y. You're never doing that. You don't pull the same allele. You're doing big Y, big R, little y, big R, big Y, little r, little y, little r. Every year I have students get that wrong, but that's how you set up the dihydroid cross. And now, with the time I have remaining for my conference, I'm going to go and show how to do the pre-Punnett skills worksheet. Sheep. Bah. Okay, see you guys later. Thank you.